Hi everyone, my name is Danielle Soileau. My mentor is Dr. Kucknoor, and my objective is to test if a TV homologue TV81 is present in T fetus and if it will play a role in promoting adherence to bovine epithelial cells. So what is T fetus, you may ask? T fetus is defined as a microscopic single-cell parasite that has traditionally been identified as a cause of reproductive disease in cattle. The rationale behind the proposed research focuses on the idea that the mediation of host cell adherence could be exploited for treatment regimens or vaccine strategies. If T. vaginalis, which causes Trichomonas in humans, has been found to possess a novel cell surface protein, TV81, that edits the binding of the parasite to human epithelial cells, it is plausible that similar yet to be identified genes exist within T fetus genomes. It was known already that TV81 expression goes up with a higher endurance, aka it is a sticking population. Our goal was to test if this homologue was present in TF to see if that gene was present in the genome of the cell. We had to take that out of genomic DNA. Next, we did a PCR process using primers based on TV81 genes. This allowed us to synthesize the primers first. Then we used them to amplify that from the genomic DNA. On the gel, we amplified TV81 and TF. We tried TFK and TFF, but we only got TFF6 and TF31 to work. We had 2 samples of TFK. The older one worked, and the newer one didn't show the results we were hoping for. Thus, we ran a PCR for those to better understand why one worked and the other didn't. This process is known as gene amplification. It is an amplification from TV81 based on primers that were designed on the sequence of this gene from a genome database. This process is known as genomic DNA PCR. In the second test run, I ran different samples of TF31, TFK, TFF6, and TV123 as a control group. While the TFF6 samples seemed to be degraded, I was pleased to see the TFK band worked. This encouraged me to use TFK, TFF6, and TF31 throughout the rest of the experiment. RT-PCR, the second experiment that we did, is defined as a reverse transcription polymerase chain reaction that is a laboratory technique combining reverse transcription of RNA into DNA and amplification of specific DNA targets using polymerase chain reaction. It is primarily used to measure the amount of specific RNA. We used TF31 uninduced and induced as the experimental group, and tubulin uninduced and induced as the control group. If TF31 has had any contact with HeLa, we call it induced, and if the TF31 hasn't come into contact with the HeLa cells, we call it uninduced. For the TF31, we saw a nice band for the induced and a faded one for the uninduced. For the tubulin, whether it was sitting on HeLa or if it was by itself, it served as a skeleton. Tubulin, the housekeeping gene, will not change. This is shown in the picture above. This proved that TF31 also has the newly identified TV81 gene in Trichomonas and is present in Tritrichomonas. We also observed that the gene is increasing expression when T fetus cells come in contact with host cells. This makes sense, assuming that the function of TV81 is adherence. The cells were trying to bind to the host cell to make more proteins. In the final process of the project for the summer, we chose to run an antisense oligo. During the antisense oligo process, we had a control group which contains DNA, makes RNA, and ends up with a final product of proteins, and then the treated Trichomonas with oligo. We did a few RNA isolation processes and ran another RT-PCR process. We used cells that were both 24 hours old and 48 hours old to see which would show up the best. Often we had better luck with the freshest samples, the 24-hour ones. In the RT-PCR amplification on the gel, we saw a small reminiscence of bands in the treated area and nothing in the control groups. The goal was to knock out the expression. I found this process to be one of the most interesting, even though it was difficult to capture the gel's band on camera. I was taught how to use an electrocell manipulator, which is a pulse generator that can be used to introduce DNA, RNA, and a variety of other molecules into prokaryotic or eukaryotic cells of interest. The unit can also be used for cellular fusion techniques, as well as other procedures that require a controlled electric pulse. Full regulation of voltage, capacitance, and resistance coupled with the exponential decay system allows for unparalleled control over time, constant in the precision pulse system. So the next question you may ask, where do we go from here? In the future, we can continue the project. Our next goal would be to run an adherence assay for the samples. With the control cells, we'll measure them with fluorescence. For the treated group, we will knock down the adherence protein, meaning we should barely see any adherence. By meddling with all of these techniques, we assume that we will be able to publish an article stating that TF81, also like TV, as previously published in TV81, allows TF to bind to the host cell. This would confirm our hypothesis that it helps the process of infection. Thus, we would examine the structure of the protein in order to design chemical targets to be used as treatment drugs in cattle. We plan to present at the Texas STEM Conference and at the annual Undergraduate Research Expo at Lamar in 2022, with the combined efforts of the entire student lab of Dr. Kucknoor, and we plan to publish a scholarly article based off of years of cumulative research that has been put into the study of Trichomonas. Like any path found in science and medicine, students have passed the baton of knowledge under the guidance of Dr. Kucknoor to harvest a wealth of knowledge worthy of publishing. Overall, I learned more than I ever imagined before starting the project. Not only did I learn new lab processes, I learned how to organize a professional project and how to speak more confidently in front of my peers. During my first presentation, I was exceedingly nervous. SURF gave me an otherwise not feasible scenario where I got to listen and discuss with older students on how to present a project in a professional setting. This was more vital than ever, not only for future skills, but it gave me a chance to reconnect with my peers and school after being in the pandemic isolation for so long. SURF has given me a better appreciation for research. In the future, I plan to attend medical school and eventually become a doctor. Working with Dr. Kucknoor during the research project opened a new molecular world to me. Not only did it foster new interest and help me understand topics that I had learned in class before, it has pushed me to want to continue to be involved in and supporting research projects in the future. In a world where it feels like we are currently under attack by an invisible enemy, COVID-19, it is crucial to keep exploring across the terrain not seen by the naked eye. Many of the processes I learned, such as the PCR process, is technology that was used to help fight against the spread of COVID-19. The skills I learned this summer are priceless and will hopefully be able to help me understand more diseases in the future. Thank you, Dr. Kucknoor. Thank you, Dr. Burham. Thank you to the Office of Undergraduate Research. And thank you to all of the friends that I made in lab along the way. My experience with SURF will be something I will always have a special place in my heart.